Hello, in this video we will discuss about the CRISPR-Cas9 system. So basically what is the CRISPR-Cas9 system? Basically it is the natural defense system in bacterial organisms. And the natural defense system so has recently been used to modify genomes also of the most important protozoa and parasite. Protozoa and parasite. So what is CRISPR-Cas9? CRISPR-Cas9 means the CRISPR is the stand for CRISPR is the clustered regularly interspaced. Interspaced mean the space between them. The cluster of regularly interspaced short palindromic repeats. Short palindromic repeats mean we will discuss later in this video what is the palindromic. So the first of all the natural part of genome it is, and bacteria and archaea is the major example for these CRISPR-Cas9, and it provides adaptive immunity against viruses and plasmid by using CRISPR RNAs. CRISPR RNA that silence invading nucleic acid, foreign nucleic acid, which for example the bacteriophage. So remember the CRISPR-Cas9 is a part of the adaptive immune system in the bacteria, same like a B memory cell and B cell and T cell in the human body. So here is I am drawing the bigger picture of Escherichia E. coli, which that is contained here is the plasma membrane, cytoplasm and membrane, and the pili. On the other hand, this is the bacteriophage. This bacteriophage is contained DNA. This DNA is basically, here you can understand, this DNA will be injected after the landing to penetration to contraction to transformation. After this transformation of this DNA, which that will be fixed, here is the plasmid contain Cas gene and the promoter as well as a CRISPR locus are present, which that contains spacer and the repeat. The spacer is the different colors while the repeat is the similar color. So the palindromic repeat. On the other hand, here you can see this is the DNA of the bacteriophage, which that will be bound to form a new spacer. After this new spacer, it will be replicate to produce RNA. So remember, so here is the Cas gene. This Cas gene will be activated to produce the CRISPR-Cas9 complex. While this is the another promoter will lead to CRISPR locus to transcribe the spacer to produce the different type of mature CRISPR RNAs. This basically the specific CRISPR through a guide DNA, a guide RNA, sorry, the guide RNA will be bind with the CRISPR-Cas9 complex. After this binding, the phage DNA. This phage DNA basically will bind, and in this way, the basically is the endonuclease restriction enzyme will cut this DNA, which that is the harmful phage DNA will be cut through a CRISPR Cas9 complex by this mechanism. So remember, this is the basically is the spacer will produce this spacer will produce the RNA look like a hairpin. So in this way, here is the degradation of the phage DNA. After the phage DNA degradation will lead to clear the pathogenicity of the bacteriophage. So what is the palindromic sequence? The palindromic sequences mean the contain spacer as well as palindromics. This palindromics contain cytosine guanine and cytosine guanine. In this way, a structure of cruciform hairpin DNA with a palindromic sequence. And in this way, here you can see the cytosine guanine with the guanine with cytosine will bind to form a hairpin. So a structure of cruciform hairpin DNA with a palindromic sequence is known as the basically mature CRISPR RNAs. So anyhow, here is successfully genome manipulation, changing the present view of genetics in parasitology and CRISPR-Cas9 system will have a significant influence on the process of developing novel drugs, treatment strategy against protozoa and parasites. So remember, against the protozoa and parasite, the CRISPR-Cas9 can be can be developed the novel drugs against this protozoa and parasite. So anyhow, it can be clear the disease also. So remember, here is the process of, again I am describing the Cas gene containing the repeat spacer and repeat, this repeat spacer and repeats. So different type of repeats, and the spacer is the different also. So repeat is same, can be, you can understand. So remember, and in this way here is the Cas9 gene, the new spacer can be added. After the new spacer when add, for example the bacteriophage containing DNA, which that after the binding the CRISPR-Cas combination, and this binding through a ligase enzyme and the restriction to ligase, and in this way here is the Cas complex, the CRISPR-Cas9 complex, which that foreign DNA will bind, and in this way the CRISPR RNAs will be bind, and in this way the foreign DNA will be bind and degradation. So in this way the CRISPR RNA which that is here, and the degeneration of the nucleotide will be occur. The mechanism of genome editing using CRISPR-Cas9. The mechanism of genome editing using CRISPR-Cas9. So genome editing with Cas9 nuclease pathogen associated molecular pattern, which that here you can see the you can understand this is a Cas9 protein. This Cas9 protein containing a single guide RNA. So this is the genomic DNA. So remember, this is the genomic DNA and this is the target sequence. This is the target sequence which that will be target the guide, a single guide RNA. So in this way here you can see the pathogen associated molecular sequence are here, the blue color, and this is the brown color showing the target sequence where the targeting of the single guide RNA. The Cas9 protein here is a complex. So after this process, here you can see this is the CRISPR RNA. So this is a single guide RNA and Cas9 protein. So in this way here you can understand the CRISPR RNA, which that is front of the DNA of the genomic DNA of the, for example, the genomic DNA, which that is here for editing. So in this way the mechanism of genome editing using the CRISPR-Cas9, through this mechanism, in this way the restriction to cut this DNA. After the cutting, the two situation can be occur. After this cutting, the two situation can be occur. In this way, the non-homolog end joining NHEJ, basically Q70, Q80, and ligase IV. In this way, the indel mutation will be occur. Indel mutation mean this will be bind here. You can understand. So anyhow, here you can see this is the HDR, the homology direct repair. The homology direct repair mean the ATM, ATR and RAP5 prime. In this way, RAP5. In this way, the donor DNA which that is required for the binding, and the targeted the genome editing. In this way, the donor DNA will be bind. For example, our gene of interest, which that can be bind. But the non-homolog end joining the NHAG is the indel mutation. So the indel mutation can be occur. But this is the mechanism of genome editing using the CRISPR-Cas9. So what is the application of the CRISPR-Cas9? Basically type of editing, also the genome editing with Cas9 nuclease, genome editing by double nicking, and visualization and localization, transcription and regulation. This is the different type of editing, while the application is the HIV/AIDS research, gene editing, prevention of the genetic diseases, basically HIV, AIDS research and gene editing, prevention of genetic diseases, and cardiovascular diseases also. So the cardiovascular diseases prevention, for example, atherosclerosis and arteriosclerosis. And on the other hand, the metabolic diseases and viral diseases, hereditary eye diseases, and cancer treatment and genetically modified organism can be prepared. So anyhow, this is the CRISPR-Cas9 system, which that I had discussed about the what is CRISPR-Cas9 and the mechanism of genome editing using the CRISPR-Cas9. So thanks for watching. Please make sure to subscribe, like and share if you like this video, and share to another, and give the comment below for understanding better, and give the question. So thanks for watching. Bye.